Hello, Priya, once again today. Now, our next presenter is the Priya DasSarma from Blue Marble Space Institute of Science, and the presentation is Haloarchaea as potential for the space travel and proxy for deciphering the limits of life in the time of the climate change. And now, Priya, the space is yours.
Well, thank you. Dziękuję. I'd first like to thank Dr. Jazowski and the organizers for the invitation to speak at this wonderful meeting. I've really been enjoying it so far, and I'm going to be talking. I think this is the first talk that's bringing in the actual bio side of astrobiology, because we actually are working with whole organisms. This is a talk by myself and my husband. We work in Maryland, USA. So for those of you who are not familiar with Haloarchaea, and I'll go back to what Dr. Osinski mentioned yesterday, we do use a lot of Greek and the term halo meaning salt, archaea meaning ancient. These are thought to be ancient microbes. And so this graph will give those of you who are new to it a little bit of perspective. So our organisms are the ones that are actually all the way on the right-hand side of the screen with the age, and you can see that their growth optimum is in environments with 4+ molar salt, which is in stark contrast to what people generally think of as salty, meaning seawater. And that's, for example, an area where you would find cyanobacteria, and that's just about 8 times lower in salinity. So our organisms are polyextremophiles, and I know that Marta gave a nice introduction to extremophiles. I don't have to give you a lot more. Our organisms actually contain the ability to survive many different extremes, not just one at a time. So the saturating salinity all the way up to 5 molar salt. They can survive desiccating conditions, so within salt crystals, like you saw on my cover slide, toxic ions, including perchlorate, which has been found on Mars. They can even use that for an energy source. They can survive low oxygen conditions. In fact, they can grow aerobically, anaerobically, and even phototrophically. So in other words, using the sunlight to generate energy. They can survive the stressors of all sorts of radiation, including UV and ionizing radiation. I'll talk a little bit about that today. And as I mentioned in the chat earlier, our Antarctic strain can even grow sub-zero, which is possible because our brine is so high in salt that it actually doesn't, our solutions don't even freeze at that temperature. So we think that they would be able to be good candidates for living on Mars, potentially, and other planets, because they're able to survive all these extremes. So again, a little bit of perspective. So we believe that these organisms have been around for a very long time, but we know that the history of Earth stretches back, of life on Earth, it stretches back 4 billion years or more, starting out with Luca and subsequent cellular life, with all of them containing basic factors, including the universal genetic code, the ability to generate energy, and membrane-bound compartments of different complexity leading up to the 3 domains of life, the archaea, the bacteria, and the eukaryotes. So I'm going to focus sort of on the Phanerozoic era, which is sort of when there's been an abundance of life. But if you focus a little bit more closely into the Paleozoic, and then look down to the Permian era, which is right about here, which is between about 300 to 250 million years ago, the Permian era was quite interesting. And I'll tell you why we are particularly interested in this at the moment. We were able to isolate organisms, halophilic archaea, from salt from the Permian era. And the Permian era is interesting with respect to climate. And if you look at the graph in the third, sort of the graph with up and down here, this is temperature. And what we're looking at, the average global temperature today, it's around about 14 degrees Celsius. Back in the Permian era, it was as high as 32 degrees Celsius. Globally, if you average that out, that's very, very hot. It isn't the Hadean era, but it was very, very hot. There was a massive volcanic action at that time. This was at the time when Pangaea which again from Greek meaning all, and Gaia from earth, and Panthalasia, all of ocean was there. There was a lot of activity going on a geological level, and there were these massive volcanic actions. There were elevated CO2 levels. They in fact went up from about 350 or about 400 all the way up to over 600. That's pretty much the trajectory that we're at today. And just to give you a little bit of perspective, the CO2 levels up until the industrial era for about 6,000 years that we can look at was right about 250 something, round about there. So we have already increased the CO2 levels. You all know about that. That the elevations were there also in the Permian era. There was greenhouse gases, there was acid rain, darkening of the sky, so lack of photosynthesis. The oceans were acidifying, they were becoming anoxic. And for those of you who are paying attention to the news, it sounds pretty similar to what we're dealing with right now. That was happening over millions of years. We're doing this in decades. So I think that it's very interesting to study something that survived those kinds of conditions, potentially, and see what mechanisms does it have to adapt, right? So we're using it sort of as a model for that to look at. So we isolated them out of halite deposits, such as this. And we've also isolated these microbes from other places. Our model organism, which we actually didn't isolate, but Walter Stachinius isolated, was from South San Francisco Bay, from the salterns there. And you can see how beautiful they are. And that's probably one of the biggest draws I had to the Haloarchaea coming into the field, was that they're absolutely beautiful and come in many different hues. Actually made the cover of the American Scientist. We're kind of proud of that. But as you can see, it came in the same issue, The Salty Side of Life. That's our article, came with Confronting Climate Change. And I think that's something really to keep in mind when we're talking about traveling in space. We're also talking about traveling in time in terms of conditions. Other places we've isolated these novel Haloarchaea from are from the Dead Sea in Jordan, from the high Andes in Salar de Uyuni in Bolivia, from the Great Salt Lake here in the United States, and you can see the beautiful pink there, and from the Tarija mines in Bolivia, and you can see the absolutely stunning colors of these Permian salt deposits. We also, as I mentioned before, have a laboratory strain that was isolated by Fransman et al. From Deep Lake Antarctica. So they're from all over the world, but they are actually an interesting monophyletic set of organisms. And if you can see here, we have the Haloarchaealis, which is where our model microbe NRC1 comes from, is situated. And then here's where HLA is from. This is the Haloarchaealis. These are this is the Antarctic strain. But all of our Permian strains all ended up being in the Natria albalis grouping here. So we thought that was kind of interesting. We've been focusing and doing a lot of genomics on them. We have sequenced over a dozen strains, and these are publicly available for anyone to look at and in detail on our public website, HaloWeb, and that link, as well as other links, have been provided to you in the chat. If you go there, you can look at the genomes of any of these organisms. For example, this is the Mayan strain, so you could just click on that and go to that. I don't have time to show it to you now, but I'd be happy to share offline. When we did the first genome, sequenced by our laboratory, led by our laboratory in 2000 and published on the cover of PNAS, we discovered several important traits of Haloarchaea, and these may be a hint at how you survive in these extreme environments, one of which is the acidic proteome. And that's really, really critical when you think about the fact that these organisms actually have a 5 molar salt concentration inside of their cells. It's potassium chloride inside and sodium chloride oftentimes outside. And this is an interesting phenomenon. We thought, okay, we discovered this in the Haloarchaean NRC1. This was the first one sequence, but we have found that with every subsequent Haloarchaeal genome, this is still held true. So this is a very important trait of these particular extreme organisms. And you can see here on the graph of the PI, you can see here that here's NRC1 and HLA, and all the other organisms that exist have either bimodal distribution or are basically they have a neutral PI on average. And here, for example, is a surface charge negative, the surface of transcription factor complexes, and you can see that here the Haloarchaea are mainly red, acidic, whereas the human one is blue and not. So we studied these organisms extensively, especially the model microbe Halobacterium species NRC1. And we've looked at many different aspects of their survival in extremes on Earth and beyond. Actually, this should be updated, this slide. So we've looked at the desiccation aspects, the heat, the cold, metals, osmotic stress, light, UV, ionizing, and different forms of respiration and fermentation, oxygen limitation. Now it's on. Okay. But one of the things I wanted to point out to you is that one of the stressors that organisms are going to find themselves in when they're going into space and when they're going into these extreme environments is the fact that there is stress of ionizing radiation and UV. And they do incredibly rapid, we did this experiment some time back, incredible rapid repair of double-stranded breaks. Here you can see after ionizing radiation, the genetic material, which was intact initially, has been sheared almost, and the fragments are only about 50 kb. But after a couple of generations, they have regenerated the DNA. They've fixed and repaired it. And it's pretty amazing. So this hints at the fact that they can survive extremes. We looked at this using a Linux machine, and we were able to find that we were able to get derivatives of our wild-type strain, NRC1, that were more radiation resistant than Deinococcus radiodurans. And I'm happy to discuss this later, but you can see that they're growing at a much, so their survival is much, much higher than Deinococcus or the wild-type strain. And our organism, as I said, was very beautiful. We've isolated derivatives that were both containing gas vesicles and not containing gas vesicles, which had originally been thought to be the factor that was protecting them. When we looked at these transcriptomically, we looked at an average of multiple independently isolated mutants. We found that the only thing that they had in common was that they had an operon of RPA encoding genes upregulated. These are eukaryotic type single-stranded DNA binding proteins. And this was very, very interesting and may very well be of interest for further work. But we thought maybe it could be a fluke that they just all happened to have done that. Well, we took that same operon and overexpressed it in the wild-type strain. And here you can see with the survival, this was the wild-type strain. When we made the derivative PDRK1, it basically had the same survival, very close to the same survival as the LH strains, the ones that I showed you earlier. So I think we've proven it. This operon is very, very important in surviving ionizing radiation. This will be important also for our astronauts going out into space. When we're talking about space, I'm not quite in space when I'm talking about the stratosphere, but the stratosphere, which is close to us here on land, if we send our microbes up there, we basically have a laboratory test site for a Martian analog without a surface to test the survival of our model microbes. And we said, you know, they can handle all these stresses. Can they handle Martian conditions? And I'm not going to go over the exact details here, but you know, the temperatures there are low. There's UV flux. In fact, over the ozone layer, there's elevated UVC. And we know that on Mars, there isn't that ozone layer. So there they would be very much exposed to UVC. There are low oxygen levels, pressure is low, it's extremely dry, and so on. So we were probably the only laboratory that's actually sent up live microbes up into space. Most of the other experiments that are all the ones I know about have been lyophilized or just spores. Ours was sent up in their media. They came back in liquid. They were obviously in a liquid media, even into the very low temperatures because of the high salt concentration. And what was amazing to find out is that when we sent them up, we had obviously got a ground control, one that was sent by plane to California. This is Death Valley, which is where the experiment was done. And those that were sent up in the balloons up into the stratosphere, the survival was pretty high. In fact, this is in the NRC1, the San Francisco Bay strain. This is the Antarctic strain, and you can see this almost complete survival. Here's our, they're here.
3 minutes.
Sorry?
3
okay,
minutes.
I'll speed up. Okay. So NRC1 was more resistant to UVC and pressure, whereas HLA was more resistant to the cold. So that makes them pretty interesting for survival on Mars. Just one more interesting fact that I thought I would share with you about our organisms is the purple Earth hypothesis, which came out of our lab. And that's referring to the way that organisms produce their energy. They use pigments that are, you can see here the retinal pigments that are found in the purple membrane of our strain. And you can see that they're using the sunlight energy at its maximum. And we believe that this was probably the original form of energy derivation on our planet, because as you can see, the chlorophylls on the left and the right of it basically had to probably just come and use part of the sunlight energy that was not being used by our organism. And, you know, they float, so it's potentially the source of energy. So, and you can see here's a purple membrane isolated in a sucrose gradient from our strains, and we think that's pretty awesome. You can check out our link on EONS. So in summary, the Haloarchaea can be isolated from Permian halite from an era of extreme climate change. We may learn something from how they have done that. Haloarchaea are remarkable microbes that survive multiple extremes. They're polyextremophiles. And we believe that the upregulation of the RPA, or single-stranded DNA binding protein, will increases their IR tolerance and potentially could be used on other organisms. And that the survival in the stratosphere suggests that these organisms could potentially survive in Martian conditions. And I did mention that HLA does. We have a paper published some time ago on their utilization of perchlorate for energy. So, and finally, that the purple Earth hypothesis suggests early evolution of phototrophy. And with that, I would like to say dziękuję. And thanks to the DasSarma laboratory colleagues and collaborators over the many decades that I've been there, as well as the many supporting institutions. And I've listed some of them here. Thank you.
Okay, thank you, Priya, for the really interesting talk. I will have some questions, but first, the questions from the audience. We have the question from Nicola. How much is known about the molecular mechanism by which these extremophils repair their DNA? Is it similar to the more modern eukaryotes?
Yes, that's actually quite interesting. I'm glad you asked that. Yes, it is very similar to the eukaryotic system. In fact, we consider them junior eukaryotes. I can show you the model that we actually have. That was actually, can you see that? Oh boy, here. So that's sort of the idea of what we think is going on. Did you want me to go into that further, or?
I don't know. I don't know. Can you precise your question, Nicola? If we have we still have 5 minutes, then maybe you can try.
Yeah, I would be happy to discuss that. I can also provide a link to the papers that we have for that, and then maybe that would be a great way to find out about it. Would that help?
Okay, maybe it will be the perfect for Nicola also. Yeah. And the next question is: are there any representatives of the Haloarchaea that have a wide range of tolerance for a certain factor?
For what?
For the certain factor.
For which factor? I'm sorry, I'm not getting.
You know, the whole is a very nice presentation. Thank you. I suppose most Haloarchaea require extreme conditions to live and have adaptations to allow them to do so. My question is, are there any representatives of Haloarchaea that have a wide range of tolerance for certain factor?
So you mean like that? Can they grow over a range of salinity and temperatures?
Yeah, I think so. That is the question.
Yes. So we can grow them over a range. So for example, you saw in the first graph that they can grow from 2 molar all the way to 5.1 molar salt. They can grow at temperatures ranging from sub-zero all the way up to 50. They can grow with different concentrations of different what we consider toxic elements. They can grow with arsenic. So they can grow over a wide range of different conditions. If I'm so sorry, I'm not sure if I answered that question right.
I hope so. That's this answer. Maybe Kaya will precise the question later. But I have the question because I'm working, as I told you earlier, I'm working on tardigrades. And you probably tested your Haloarchaea with the perchlorides, which are present on Mars. And I want to ask, what was the survivability rate of the Haloarchaea in the perchlorites, which are similar to this Martian one?
Okay. All right. So I am actually not a co-author on this paper, so I apologize. I'm going to pull it up. I can pull it up right now. Can you see my screen with the images?
Yeah.
So this is anaerobic growth of HLA and NRC1, or two model organisms, in the presence of
Okay, I
perchlorates.
will try to read the paper, because the perchlorides are in general very toxic, and
Yes.
we tested some cyanobacteria, we tested some tardigrades and nematodes, and all of these organisms, by the way, survived in perchlorides, which is quite amazing.
Yeah.
But anyway, maybe I will write you the email later. But we
Absolutely.
we have one more question from Marzena. Wonderful talk. How this acidity of proteome influence protein-protein interaction in cytosol? Do you think that there are mostly untypical from the one we broadly study now?
Right. So again, I am actually the microbiologist in the lab, so I'll speak for my colleagues, but I'm happy to provide the links to the papers that we have on it. The acidic proteome, we believe, is involved in its interaction in this high salt environment. However, we do think it's a very intriguing question, because we're looking at it binding, for example, the DNA binding proteins are also acidic. So if you're talking about a nucleic acid that's acidic and you're having a protein that's binding to it, it is a little mind-boggling how they're doing it. I don't think it's known in great detail exactly the mechanisms. We're still working on it. Even though we've known about the acidic proteome, we have not delved into it greatly. But I think it's an adaptation to the high salt, because if you have things that are just not bimodally distributed, but they all have the same charge, they're actually going to repel each other. So in a cytosol that's highly salty, that basically then you can keep them apart instead of having them salt out. And now when I'm talking about salt, I just wanted to bring up something that I didn't really discuss in great detail. We have a bunch of teaching modules and so on, because this is a very easy to grow set of organisms. They can grow in high salt. They can grow in a regular incubator. And so it's not an extremophile where you need a very complicated equipment. I think, Lukasz, in answer to your question earlier, you don't need those high pressure environments or glove boxes or anything like that, but you can grow them readily in a standard laboratory. And that's what makes them very easy to work with. And also, we have developed everything from genomic and transcriptomic and molecular biology techniques. In fact, we have a halophile manual with all of that in there, and it's available to the general public, and we're very happy to collaborate and answer questions.
Okay, thank you, Priya. We don't have time for more questions. If you will have any more questions to Priya, please contact with her.